Good morning, everyone. My name is Mary Bishop, and I will be presenting on the microgravity-induced differential gene regulation in the symbiotic bacterium Vibrio fischeri. Okay, so this morning I'll be talking about the significance of the project, the research questions, a bit of the methods, some of our results, and future directions. So my project talks about microgravity-induced gene differentiation in the symbiotic bacterium. So I need to talk about microgravity. What is microgravity? Minus g, very, very low gravitational force. And in the International Space Station, ISS, the astronauts feel this, and that gives them the weightlessness. As you can, I'm sure everybody has that picture of the astronaut flying around the ISS. And the effects of microgravity on humans and microbes are extremely interesting. There's microbes that have different gene expression just by the microgravity, just having that different gravitational force. And also the host microbe interactions are changed. So as you may know, the humans and microbes, we're more microbes than actually human. We have more microbes on our body. So it's really important for us to study these interactions. There's been a study by Dr. Nickerson, and it talked about salmonella. And what they did is and gravity conditions, then they took that cultured salmonella and infected mice. Well, as you can see from the graph, here we have microgravity right here, and the time of death in the x-axis, and then the gravity, that's the time of death. As you can see, the mice died much faster from the microgravity affected salmonella. And there's been numerous amount of studies on salmonella. We know a lot about them, but guess what? There are more good guys than bad guys. So we need to talk about the beneficial bacteria. What about beneficial bacteria? So that's the study of the project. So what is the impact of simulated microgravity on beneficial microbes? And the beneficial microbe we chose is Vibrio fischeri, and it is the model organism we chose because it formulates a symbiotic relationship with the epithelial squid, as you can see here. Vibrio fischeri is bioluminescent, and I'll talk about later on how important it is, but again, it infects the light organ located in the mantle of the Hawaiian bobtail squid, and this is important because the same human microbe interactions, they affect our epithelial cells. So it's important to study that and use the squid Vibrio model in our project. So Vibrio fischeri, it's as really how they look, but it's nice to get a picture of its bioluminescence, but it's really not that green. And this is the bobtail squid. This is a baby, actually. And this is the light. And what happens is the Vibrio fischeri actually infect that light organ. And it's useful, very useful, because what happens is in the moonlight, the Hawaiian bobtail squid, they come out, and they're looking for shrimp, as you can see here. And it's useful because predators, and the Hawaiian bobtail squid, they have a flashlight here as the picture, but it essentially is like a flashlight. And so the predator it just looks like moonlight. So they go past it, and it's great. And the Hawaiian bobtail squid actually has, I don't even think it has a predator. So it's great. It's a great symbiotic relationship. And it's a mutualistic symbiotic relationship, which means that they both benefit. So Vibrio fischeri has a great place to colonize and just grow and be happy. And also So in this project, the specific objectives is to examine the changes in the Vibrio fischeri transcriptome in the presence and the absence of gravity. And we have two specific objectives: to assess the role of the global regulator, HFQ, and I'll talk about these things a little bit later, in microgravity response. So once we get that data back, we can use the microgravity and the to compare, just like the other study did. They compared it side by side, microgravity and gravity. And just to refresh everybody, so that here's DNA, and in order to transcribe, it goes to RNA, and this is almost like a message. So we're very interested in the message that lies in this RNA. So the experimental design, we have 2 strains of Vibrio fischeri. We have a wild type, or the normal, or the parent strain, and a in HFQ. So we have 1 wild type HFQ. We have also 2 time points that we're taking. We're taking a 12-hour and a 24-hour culture. And then finally, we're doing treatments to those 2 strains at those 2 time points. Simulated microgravity and gravity as the control. So the HFQ gene, why are we interested in it? Well, it's a global regulator that is present in about 50% of all bacteria for an RNA binding protein. So here's HFQ protein. So it binds to mRNA, this protein, and it helps translation occur. And in the microgravity conditions, the HFQ gene has been shown to be downregulated. So it doesn't express a lot in microgravity conditions as opposed to gravity. And the role in symbiotic associations in HFQ is also affected by microgravity. So you're probably asking yourself, how can they simulate microgravity? That's impossible. What are they going to be in the ISS? No. What we have is this high aspect ratio vessel. And I'm going to describe it a little bit, and then I've got a video so you can really understand it. So it's simulating microgravity, so 10 to the minus 6 g-force. And what happens is this is the microgravity, and it's essentially perpendicular to a table, and it's constantly rotating, constantly changing, which is exactly what happens in ISS, which is why astronauts float. So there's a constant rotation, gravitational vector is changing, and in the gravity control, which you'll see in the video, it's on the table. It's directly on the table, so gravity is constantly coming down. Let me see if this works. Okay, so as you'll see, it's turning. This is, again, perpendicular to the table, and here's the gravity right on the table. There's no perpendicular association there. So these are our 2 treatments. Again, this is a cell culture, so we've inoculated this with Vibrio fischeri, and it's a certain amount of cells that we have in there, so we know it's only Vibrio fischeri. We have an overabundance of Vibrio fischeri in there, so we know there's no other microbes growing. So once we have the cells, we flash freeze them with liquid nitrogen. That was my favorite part, I think. So we have Vibrio fischeri cells. We have the wild type, the parent strain, and the strain with knockout of the HFQ gene, which I talked about earlier. So we take these flash frozen cells, we extract the total RNA. So there's a lot of RNA. There's mRNA, which is the message. There's the transfer RNA. You have ribosomal RNA. So we're just getting all of the RNA at this point. We then test it for both quality and quantity, and I'll talk about how we did that. Later on, we deplete it of the ribosomal RNA. We're not interested in the ribosomal RNA because we want that message. We want the messenger RNA. Then we convert it to cDNA, so we go from single-stranded back to double-stranded. Then we sequence it, just like Fernando will eventually do, but he's interested in the metagenome, but we are interested in the transcriptome. What's happening at that very moment where microgravity and gravity conditions. So here are some of our results. This is a histogram from the Quantitative Pcr. Again, Fernando did PCR, which is amplifying, so it's making copies after copies of the DNA, whereas quantitative PCR is taking pictures of what's happening in that very moment. So it's very interesting, especially when you want to test for wild type. So we have the wild type RNA, the HFQ gene, under gravity conditions, it's upregulated. This is the normal expression. As you can see, under microgravity conditions, it's downregulated. So this, you can clearly see the difference between what's happening under gravity and microgravity conditions. And this has been tested, and a publication from this. And this is also a way to see that we extracted high quality RNA. Because in order for the results to happen in the quantitative PCR, for it to be converted to cDNA, to DNA, it would not work with low quality RNA. So this is a quality checkpoint. We have 2 quality checkpoints. The other one is done on the bioanalyzer. And this is very similar to, again, Fernando's gel. It was a picture. It was very similar to this. It's very similar in that it uses the voltage gradient to separate molecules using by size. And RNA and DNA are both charged particles. So they separate based on size. And you can see here this is 23S, which is the large subunit of the ribosome. And then you can see here the bands. This is the 16S bands. This is another quality checkpoint because if you can see, these are nice clear bands you can see, whereas this lane 3, its degradation is present here. So we essentially will not use that to convert to cDNA. We cannot use lane 3. And that's another quality checkpoint. Not just quantity, we have to focus on the quality of the RNA. And the main point of this project is to generate libraries of cDNA in order to sequence. So here we have our 6, and potentially, Lexi told me, 10 more libraries. So we're very excited about that. And as you can see, we have library 1 has a time point, a strain, the day it was pooled, and the treatment. And then this is how much cDNA we have actually made. So eventually this table will be all full. We'll send it off to be sequenced. Then we can compare the data, just like Fernando will eventually do. So the summary, what we did is we extracted high quality RNA, it's very important, from Vibrio fischeri, the wild type, the HFQ, knockout HFQ. We converted it to cDNA, and then eventually we're going to sequence that, and using comparative bioinformatics of the 24 different transcriptomes, we're going to see what happens under microgravity conditions. Thank you very much. Oh, and I would also, I would love to acknowledge JDEEP, the NASA Florida Space Grant Consortium, JDEEP, Srila, also Space Florida and the Space Life Sciences Labs, Tony and Ryan. The Foster Lab, of course, Dr. Foster, thank you so much. Lexi, partner in crime, Diana, Giorgio, Giovanni, Kim, Rochelle, everybody helped, and this experience is life-changing, really, and I just want to thank everybody. Thank you so much. Go ahead, Johnny.
I have a question. On your qPCR slide,
Yeah.
I'm curious to know, you have the upregulation of HFQ and gravity.
Yes.
Gravity. Why is that important? Why is the upregulation in gravity versus downregulation important for HFQ?
The downregulation is important because what happens when HFQ is downregulated, you can equate it to a floodgate of virulence. So it becomes more virulent, and as you may know, the virulence, it's almost good bacteria turn bad up in space. So that's why it's important. And we can also, later on, we can also find housekeeping genes, which are important in qPCR, quantitative PCR. We can find those housekeeping genes to do further studies. Fernando.
How do you go about separating the mRNA from the rRNA?
Okay, well, we treat it with, it's a protocol. We deplete the RNA completely, and it's just using various reagents, and eventually all we'll have is the messenger RNA.
So do you use like a column to just pull the RNA while the mRNA flows, or do you?
It's various. We use actually their magnetic beads to separate that,
Alright.
and then I'm sure you use this technique of precipitation, and
Alright.
you separate it that way,
Oh.
spinning it down and
Alright.
separating it that way. Okay. Thank you.
Mary Bishop. Yeah, there you go.
As soon as this takes a little bit to get over. Ready? 1, 2, 3.